Hello everyone, welcome to today's webinar. I'm Linda Wang, Senior Correspondent at Chemical and Engineering News, and I will be moderating today's event. The presentation today is being sponsored by Wire Technology, a leader in instrumentation for determining the absolute molar mass, size, charge, and interactions of macromolecules and nanoparticles in solution. These tools include inline multi-angle static light scattering, high-throughput dynamic light scattering, electrophoretic light scattering, differential refractometry, differential viscosity, field flow fractionation, and automated composition gradients. During the presentation, we will hear from Dr. Christoph Johann, who is Manager for Eclipse FFF products at Wire Technology. Dr. Johann earned his PhD in 1985 in physical chemistry at the University of Mainz. He has been active in polymer and biopolymer analysis for over 30 years and has several peer-reviewed publications in the field of macromolecular characterization. In 1991, he introduced commercial field flow fractionation systems in Europe. In 1993, Dr. Johann founded Wire Technology's main European subsidiary. Dr. Johann has been active in the development of Wire's Eclipse FFF instrumentation, which was introduced in 2002, and has shepherded it through several generations of technological breakthroughs. I will now hand the presentation over to Dr. Johann.
Many thanks for the moderator for the kind introduction. In today's webinar, I want to discuss with you how we can get more information with less effort analyzing complex nanoparticles using FFF. FFF has been around for a long time, and I'm sure most of you have heard of FFF or have even own experience using it. There's a growing acceptance of FFF, and for example, this has motivated ISO to publish a new standard with the title Nanotechnologies Analysis of Nano-Objects Using Asymmetrical Flow and Centrifugal Field Flow Fractionation. That is a big step forward to making FFF an accepted and standardized method comparable to SEC. Still, FFF has a reputation for not being easy to use and needing an expert to get the most out of it. Today, I'm presenting a breakthrough of new developments that make the full potential of FFF accessible to a wider user base. It is a combination of new instrumentation plus sophisticated software. It provides a comprehensive set of data on complex samples that no other single method can provide. I will discuss exclusively Flow-FFF today because in the family of FFF methods, this is the most universal and widely used. 90+% of systems of FFF are Flow-FFF systems. So for simplicity, when I say FFF, what I mean is Flow-FFF throughout this talk. So before we go on discussing FFF, let's first define what we mean by a complex sample. What is a complex sample in our context? A sample which is not homogeneous, but consists of various components with different sizes and properties, at least one being a nanoparticle. Some examples of popular nanoparticles are shown here. They cover a size range from a few nanometers to about a micron, and they can differ widely in geometry, chemical composition, and structure. Each of these types of nanoparticles typically has a size and structure distribution. Now consider adding other macromolecular compounds like proteins or polymers. Then you get a zoo of sizes and shapes all in one sample. Gene delivery, cancer vaccination, drug delivery applications, sophisticated industrial applications, they all depend on complex samples to be functional. Analysis becomes challenging, and conventional methods are not able to provide the required information to optimize the synthesis and to understand how the products work or why they do not work as expected. So what makes FFF the method of choice for complex samples. Size is the most important property of nanomaterials, which makes them different to smaller molecules. To illustrate what we are talking about, this dot here represents the size of a peptide. Here we have an antibody, larger, but still in a size range that can be characterized pretty well by columns, like HPLC or SEC columns. Now this here represents a virus, and we can already understand just by looking at the size difference that columns may not be the best method to characterize them, and we need something else. And here FFF comes into play. FFF is a method that can separate these large structures and, even better, as well smaller structures like antibodies or peptides in the same sample. So what are the advantages of FFF? Let's look at that in more detail. What is the advantage of field flow fractionation? FFF physically separates our sample according to size, and therefore it is possible to collect fractions. It has high resolution compared to dynamic light scattering. It has a wide separation range, and this combination makes it powerful for complex samples. It is gentle, has low shear forces, and therefore non-destructive. FFF can be coupled online to MALS, which provides absolute molar mass and size of eluting fractions. With our new development, the Eclipse mobility, we can determine charge and charge distribution by using electrical flow-FFF, an easy upgrade to an existing Eclipse FFF system. Last not least, our new developments make FFF more robust and easier to use than ever before. To achieve ease of use and robustness, we have concentrated on 3 features: a seamlessly integrated and automated system, extended data processing capabilities, providing molar mass, size, shape, conformation, plus charge and charge distribution, and software-supported method development. After a recap of the theory, I will show you how each of these features have been implemented with the Eclipse FFF. How does Flow-FFF work? The sample is injected into a thin ribbon-like channel, which has a porous bottom wall. Part of the flow, passing through the channel towards the outlet, permeates through the bottom, creating a crossflow. This crossflow concentrates the sample towards the so-called accumulation wall, the bottom wall, by exerting a friction force. Brownian motion acts as a counterforce, working against the concentration. So the molecules are constantly moving up away from the bottom wall, driven by Brownian motion, but they will be pushed back from above by the crossflow. In a stationary equilibrium, this creates a particle cloud with an exponential decrease of the concentration as a function of the distance from the bottom wall. It is exactly the same mechanism which leads to the decrease of air pressure if we rise above sea level. In our case, each particle size will have a different average distance from the accumulation wall, which is given by its diffusion coefficient. Smaller particles with higher diffusion will be higher up in the channel, and the other way around, bigger particles will stay closer to the accumulation wall. To give an idea of the dimensions, the concentration drops from the maximum value at the accumulation wall to 0 within about 10 microns up into the channel. The typical height of the channel is 350 microns. So, the sample does stay close to the wall throughout the separation process. If we consider the transport along the channel towards the outlet, then smaller particles with more extended clouds will move faster. This is because the laminar flow profile has a much higher transport velocity as we go away from the accumulation wall up into the channel. Therefore, the smaller particles travel faster, will elute first, followed by the bigger ones. And this results in a separation. We can describe this mechanism in a quantitative way with the retention equation. The equation we see here tells us that the retention time T sub r depends on the channel height W squared and the ratio of the cross-flow rate divided by the outflow rate. The equation can be used to tune the separation by adjusting this ratio and to program it during an experiment to achieve the desired resolution. The equation also tells us that we can extract the diffusion coefficient from a measured retention time if we know the experimental parameters. It is interesting to note that the length or width of the channel do not go into the equation. That means we can produce the same fractionation in multiple ways on different geometries by adopting the flow rates accordingly. Now, let's turn to MALS as a detection method for absolute molar mass and size. How do we obtain molar mass and radius using multi-angle light scattering? The schematic shows the principle of the instrument setup. A laser beam passes through a cuvette with our solution. The interaction of the light with the electrons of the sample creates scattered light, which is measured by the detectors. They are placed in a reed head around the scattering center, and in our instruments we have 3, 8, or 18 detectors. The instrument is calibrated with toluene, a scattering standard, so we get absolute values with no reference to a molar mass standard. The scattering intensity is proportional to molar mass and concentration, and from the decrease of the intensity with angle, we can extract the size. As in the case for FFF, we have a simple equation telling us how to calculate molar mass and size. Here is the simplified equation for dilute conditions, which is valid for separation processes. On the left-hand side, we have the experimental quantities. K star is the optical constant. It has the refractive index of the solvent, the vacuum wavelength of the laser, and the d/c squared of our material we're looking at. C is the concentration, which is measured by RI or UV detectors, and R sub theta is the Rayleigh ratio, the scattering intensity measured by MALS in absolute units. To calculate the molar mass and size, we plot the concentration divided by the scattering intensity versus angle. From the intercept of the regression line, we get the molar mass, and from the slope, we can calculate the mean square radius. The result of our MALS measurements is molar mass and size for every slice of the fractogram. Now let's move on and look at a real-life example of a complex sample and see which information we can get from FFF-MALS. Here is an example taken from literature. Human polyoma VLP particles were developed for gene delivery. The virus particles are created in a fermentation process. Then the virus shells are disassembled to clean them and purify them, get rid of DNA and other cell fragments that are unwanted. And then the virus has to be reassembled again. At the same time, the cargo, in this case a DNA plasmid, is packaged inside the VLPs. But this process is not without complications. The figure we see here shows a fractogram from the Eclipse FFF separation and the calculated molar mass and size of two populations. The later eluting peak is the desired VLP particle, identified by the correct size of 20 nanometer and the molar mass of 17 megadaltons. The earlier eluting peak is an undesired byproduct which consumes valuable shell protein. This publication describes how the FFF-MALS analysis helped to optimize the fermentation and purification process and to increase the yield of the target VLP particles. The publication is 10 years old, but the results are even today impressive and the article very educational. I want to show you in the next slide what we have done in the meantime to enable new and unexperienced users to develop a separation method for similar complex samples with ease and to operate the system with high productivity. Key to this is our new software platform that we call Vision. It integrates the complete workflow from method development to the final result. All applications are started from this hub. I will only show and talk here on Voyager, which is the software for running experiments, and Scout for method development and for data processing. Here are the 4 steps to develop a method with the Scout. At first, we define a series or set of sizes which represent our sample. In our example, we enter 3 different sizes with hydrodynamic radius of 11, 23, and 51 nanometer. We can also add the temperature, the solvent, and we could also simulate a shift in retention time using a mobility if we wanted to. Second step is to select a channel geometry. So do we use an SC channel or longer channel and the spacer height? And the third step is to develop the flow program until we have a satisfactory separation. What we then do is we export the method to Voyager and run it. To illustrate the process better, I show here how Scout calculates the fractogram that we see in the figure in the center of the software based on the flow program seen on the right hand side. So if we're happy with the separation and the resolution, we can export the method and save it to disk so that we can later import it to Voyager and run our sample. Here we are in Voyager. So what we do is we import the method that we have developed with Scout. So we just select the data file, load it into Voyager, and then save the method. Next, we define the sequence table. We call up the sequence table form, enter the method that we want to run, both for FFF and for Astra, so we can read in the Astra methods here from Voyager without having to access Astra. We complete our sequence, start it, and the samples will be run one after the other. And in this one view in Voyager, we see all of the information. We see the Astra basic collection screen, and we see all the system parameters and traces that we collect from our HPLC instrument. Let's see next how the simulation and the experiment compare. Let's look at a comparison of simulation and the real experiment. So what we see here is the simulation that we used to run the experiment in the previous slide. This is the experiment done with the method, and you can see that the agreement is really excellent. The retention time matched perfectly. What we do see is the earlier eluting peak, the smallest size, is broader in the experiment compared to the simulation. The simulation assumes a monodispersed sample just with one size, and from the shape of the fractogram in the experiment, we can see that there's a size distribution here. And of course, MALS confirms that observation. Now let's see how we can use Scout to optimize the experiment after we have run it. This is what we see here. This is again our experiment. We see the crossflow gradient that we used to run this experiment. And now what we want to do, we want to increase the resolution between these two peaks. They're not fully baseline separated. So how can we do that? We use Scout to modify the crossflow gradient and to predict what it will do to our fractogram. So what we do, basically 2 changes. So we're reducing the crossflow from the higher value to a lower value, and we also then decrease it over a longer period of time, and we will stay at a higher crossflow at the end of the gradient. All of this predicts that the 2 peaks, later eluting peaks, will be more than baseline separated. This is the green trace that we see here in the fractogram. Again, this is the comparison of the simulation and the experiment. So here we see an overlay of our original experiment and the simulation of the improvement after changing of the crossflow. And what we see here is an overlay of the two actual experiments, the initial one and the optimized one in green. And again, we can see that there's a pretty good match between the simulation and the experiment. So the simulation is really useful to allow us to achieve the results that we want to achieve. Before we can calculate a size distribution from FFF retention time, we need to calibrate the channel. Calibration can be done in two ways. Either a channel height calibration, which assumes that FFF theory is fully applicable to our sample, and we use then only one parameter, the effective channel height. Second option is running a set of calibration standards. This is what we see here, 3 peaks with known size and the calibration curve on the right-hand side. It does look familiar, as it is the equivalent of an SEC calibration using standards. Scout allows us to use both the schemes and compare the resulting size distributions. Let's now see how we can trace the performance of our FFF system. All data and system traces are saved with the data file and can be viewed and compared using Scout to verify validity of the experiments. Here we have the measured crossflow versus time. It looks like one curve, but it is an overlay of 20 runs. The Eclipse instrument has a very high reproducibility of crossflow, which is critical for high precision measurements. If something would go wrong, the plot of system parameter traces allows us to identify possible problems or outliers at a glance. This completes our first tour of Vision with Voyager for running experiments and Scout for method development and data processing. Let us summarize what we can get from the Eclipse FFF experiments. On top of absolute molar mass and size from MALS, this is the list of parameters we can get from vision and mobility. So it is the hydrodynamic radius from retention time, and by adding the electrical field, we can determine electrophoretic mobility values, which can then be converted to zeta potential. Conductivity, pH, and temperature are measured in real time and provided as data traces. Let's now move on to discuss how we can determine electrophoretic mobility by applying an electrical field in our channel. We call this method electrical flow-FFF. By adding 2 electrodes to the flow-FFF channel, we are able to apply an electrical field parallel to the crossflow. After focusing the sample with crossflow alone, we add a voltage in the elution step, where the separation occurs. The 2 fields superimpose, and the retention time of a sample which has an electrical charge will change. From the shift in retention time, it is straightforward to calculate the electrophoretic mobility. If we choose the polarity of the field such that we have an attractive force towards the accumulation wall, that is, a negative charge on the particle and a positive charge on the accumulation wall, then the retention time will increase, proportional to the electrical field strength. If we have species with opposite charge, we will see one fraction moving to shorter retention time, and the opposite charge moves to longer retention time. If we apply a series of increasing electrical field strengths in several experiments, then the retention time will increase in proportion to the electrical field strength. Here is an experimental result showing just that. a systematic shift of retention time of 3 polystyrene latex samples with increasing electrical current. This example illustrates how we can calculate the charge on several components in the sample simultaneously. This is a capability which a batch measurement in principle does not have, like a conventional zeta potential measurement using light scattering. Let's now see what EAF4, the electrical field flow fractionation, can tell us about proteins. Protein charge depends on conformation and of course on solution conditions like pH or ionic strength. We can use EAF4 to measure the charge of proteins with very little effort. Here we see the result from a therapeutic antibody in the native state. At physiological pH, it has a slightly positive charge. So we can see the electrophoretic mobility is in the order of plus 0.4, corresponding to a zeta potential of only 7.6 millivolt. But what happens if we stress this antibody? Then we get a completely different picture. The charge becomes much more positive. Molar mass and size do not change for the monomer peak, but it is interesting to note that the peak shape of the monomer becomes more symmetric by adding the repulsive force on the accumulation wall. That means adding a positive charge on the accumulation wall. You can see the red trace is more symmetrical. This is the highest current that we add compared to the blue trace without adding a current. And we can quantitatively determine the charge, and now we see something like 53 millivolt zeta potential. That's a dramatic change. These promising first results indicate that we can use charge measured by EAF4 as a parameter to identify stress on the antibody that we cannot necessarily see in a change in molar mass. I have one more example of the impact of FFF in biology and medical research. Recently, a paper published in Nature Cell Biology showed how AF4 allows to identify subpopulations in exosomes, which could not be separated before with other methods. This is significant because each of these subpopulations has a different biology function and carries a specific fingerprint of proteins and biomarkers. AF4 was able to separate the fractions, and MALS with embedded dynamic light scattering was used to identify the fractions by size. The fractions were collected and a comprehensive identification of the proteins and DNA performed. This is the result from the publication, where we can see the 3 exosome populations P2 to P4 and the size measured online by dynamic light scattering. Here, a correlation function of one slice demonstrates that at high enough concentrations, DLS will give us excellent results. Here, the electron microscopy pictures confirm the different size and morphology according to fraction. A direct proof that the separation worked well and the size determination from DLS is valid. At low sample load, dynamic light scattering will not give us satisfactory results. MALS, the static light scattering, is 100 times more sensitive than DLS. Here is an example of the same exosome with lower injection amount. MALS still gives us exact sizes under the more dilute conditions. But we can extract more than just size by MALS. Here we calculate the number of particles or the number density from MALS for each slice of the eluting peak. If you look at the dynamic range, both in numbers calculated, spanning 4 orders of magnitude, and the size ranging from 30 to 100 nanometer in radius, this demonstrates the capabilities of MALS for the counting task. The FFF-MALS DLS system is a powerful platform for exomer isolation and characterization. Over isolation and fractionation by size, we get a set of biophysical parameters online. Size, both mean square radius and hydrodynamic radius, giving us insight to shape. The distribution of size as a function of concentration, charge, and zeta potential from eclipse mobility, and protein, lipid, and RNA-DNA content from Astra's protein conjugate module. At the end, let's go back to Scout software and data processing. Let's see how easy it is to process the data and produce the mobility values. We have already created a template which contains the parameter for data processing, so we load the template. After that, we load the data file into Scout. And from here on, everything works automatic. The baselines are set, the peaks are detected, the peak maxima are determined, and we see here the shift of the peaks is analyzed and then plotted on the right-hand side against the electrical field, and from the slope we directly obtain the electrophoretic mobility. So we imported not just the data for one sample, but for a full sequence, here in this case for 20 runs in one step. And of course, we can also confirm the high reproducibility of our measurements this way. The results are presented in a clear table, giving us hydrodynamic radius, diffusion coefficient, electrophoretic mobility, and net charge and zeta potential. So in this webinar, I discussed with you the Wyatt Eclipse Flow-FFF system as a powerful tool to characterize complex nanomaterials. It is based on efficient separation coupled to online molar mass and size measurement with a Wyatt-MALS detector. The Eclipse mobility allows to determine charge and charge distribution in complex samples. The system is fully integrated with a new software vision, and it provides a seamless workflow from method development to final result, and this is what we call FFF 2.0. We have seen application examples for polymer latex, for proteins, and for exosome isolation and characterization. Thank you very much for your attention.
Thank you, Dr. Johann, for your terrific presentation. Now we begin the Q&A portion of this webinar. First question I have here is, can you elaborate on how you calculated the net charge of latex and proteins?
Can you please repeat the question, sorry?
Yes, the question is, can you elaborate on how you calculated the net charge of latex and proteins?
Okay, well, to calculate, we measure directly the electrophoretic mobility. So that's the direct output of the measurement, very similar to what you would do with the electrophoretic light scattering method. So in order to calculate net charge and zeta potential, you need to know the size of the particle, which we measure either with dynamic light scattering, or we take it from FFF retention time, or and then we use the usual models that you would also use with pulse measurements. So we basically calculate the net charge and the zeta potential based on size and the ionic strength of the solvent.
Great. Here's the next question is, what is the lower detection limit for the number of particles that can be counted?
Yes, that depends a lot on the signal to noise of the light scattering measurement, because the number we calculate needs a high precise light scattering signal. So, and also, of course, the smaller you get, the larger the numbers become. And that also is a limitation on the accuracy and precision of the measurement. So I would say that maybe you can go down to something like 30 nanometers or 20 nanometers, but not much lower. Then of course it becomes more difficult.
Okay. How will the samples need to be processed to inject into the ribbon?
Excuse me, can you please repeat it?
The question is, how will the samples need to be processed to inject into the ribbon?
The sample preparation and processing is like you would have it in field flow fractionation. That means you need a stable suspension or solution of your sample. The big advantage of field flow fractionation is that you can also have colloidal material or bigger aggregates in your sample, which you would need to remove if you wanted to inject on a column, which is not necessary in field flow fractionation. So typically, what you should do is remove oversized particles that are, let's say, above 10 or 20 microns, for instance, by a mild centrifugation, but there are no other special sample preparation steps necessary.
Okay, great. Another question I have here is, can we upgrade our Eclipse with the mobility module?
Yes, that should be possible. If you have an Eclipse 3 Plus or newer, then it's an easy retrofit. So you can add the mobility controller box and the channel, and the software comes with it, and it just adds to your existing system and gives it the capability to measure electrophoretic mobility and use electrical field to modify the separation.
Okay. Can you comment on the upper size limit for charged particles?
Well, the upper charge limit really depends on what we can separate in normal mode in field flow fractionation. Because once we reach very big particles, steric effects come into play, and then the shift in retention time probably will not be good enough to calculate a reliable electrophoretic mobility number. So again, it depends on the sample and experimental conditions, but as I would think up to 400 nanometer radius should work, but not bigger.
Okay. Is there a physical separation from the sample? Can you collect fractions?
Yes. This is generally a property of field flow fractionation. You're physically separating your sample into fractions of a certain size, and you can collect fractions and use them for further offline analysis methods like electron microscopy. You can also use a larger footprint channel that we call semi-prep, and you can inject larger amounts on a bigger footprint channel to generate more material in a given size fraction that you can collect.
Okay. I have a question here that says, how high can we increase the molecular weight of polymeric nanostructures with AF4-FFF method, normal light scattering?
How can we increase the molar mass size? I don't quite understand that question. So what we can do, we can measure the molar mass and the size. But what's the question? How can we increase the molar mass?
Yeah, the molecular weight of polymeric nanostructures, the AF4.
Yeah, AF4 separates. So I don't quite understand that question. So we can separate. We can also, of course, if you have, let's say, a colloidal particle, nanoparticle, you're adding a polymer. So you put it on the surface so that structure becomes bigger. We can separate the uncoated and not coated particle, and we can see a shift in retention time and also see a change in charge if we modify our sample by, for instance, adding a polymer on a particle. I don't know if that was the question. So we are able to both separate and also characterize a change in size that happens by interaction of a particle with the protein or with the polymer.
What volume of sample is typically required?
Well, we are very variable in the volume. So in asymmetric flow-FFF, you can inject small volumes like a microliter or something that your autosampler can still handle. But you can go also up to very high volumes, like hundreds of microliters of volume. If your sample, for instance, is in a diluted state and you want to inject a larger volume to have enough material for your analysis. It's more, let's say, the amount of sample that is critical in order to allow us to get a good detector signal. So it's more that we need for certain molar mass or size range, a certain amount of sample. Typically, we talk about microgram amount of sample that we're injecting in a very variable volume.
Okay. Can we use organic solvents such as chloroform, tetrahydrofuran?
Yes, this is possible. So there's an organic version of the Eclipse, so it's compatible with these solvents. Also, alcohols are possible, acetone or so, it's no problem. Of course, in non-conducting solvents, we cannot use the regular mobility module because we need a conductivity in the solvent to measure electrophoretic mobility. But for separation, it's easily possible to use organic solvents.
Okay, getting back to the other question about data 4 that you were confused about, the participant wrote in and clarified. They said, my question is, what is the upper limitation of molecular weight of particle by measuring through FFF?
Okay. So again, the upper limit, we have to distinguish between separating, the separation and resolution on the one hand, and the detection and measuring of the size by light scattering on the other hand. In terms of separation, you can go up to something like a couple of microns. Let's say 1 to 2 micron, I would think is the upper limit for normal mode FFF separation, and I would say also a practical limit in the use of FFF. Also in terms of calculating the size by static light scattering, that depends a little bit on the sample and the refractive index of the sample. We can also go up and calculate sizes up to a micron or 2 microns. So basically we are covering the complete sub-micron size range. Once you reach a size above a micron or higher, then you would use other methods to characterize your sample.
Does the shape of the particle affect the size measurement?
The shape of the particle certainly influences the resolution and the separation because that depends on the diffusion coefficient which goes into the separation. So with static light scattering, we can measure the mean square radius. That means we get a size parameter that is also an indication of the conformation and size if we can relate it to the hydrodynamic radius. That means we can measure the size, and if it's not a spherical particle, by combining static and dynamic light scattering, we get an indication if we have an elongated particle, for instance, a more rod-like particle, or if we have a compact structure. So our measurement is not depending or assuming a certain size, a certain shape, sorry.
Questions. What is the expected baseline resolution for nanoparticles? For example, can you get baseline separation between particles with a 10% difference in size? How does this vary over a size range?
Yes. So the resolution, typically, I would say we can get baseline separation if the size is different by something like 30 to 40%. We certainly cannot separate two populations that are 10% different in size. This is outside of the resolution capability of flow-FFF. But we can certainly, if you have a mix of sizes or a continuous or a polydispersed mixture, we can see the increase of size with retention time. So we won't resolve individual peaks, but if there's a mixture, we can see how the size increases from fraction to fraction, and there the resolution between two eluding fractions from the channel is in the order of 1 or 2 nanometers. So if we measure something like a tube latex sample that has a very narrow size distribution, we can still resolve that at the beginning of the peak, the size is something like 4 nanometers lower than at the end of the peak. So we can resolve how the radius increases within the peak.
Can you do an analysis of a mixture of organic and inorganic
solvents? That is possible. You can. Yes. So the question was about, can we run with mixtures of organic and aqueous solvents? That is possible. You can do that. You have to be a little bit careful because you're running into problems with detector signals. You cannot use a refractive index detector as a detector anymore because you will get shifts in refractive index because there will be a certain change in the composition during the analysis, but you can use light scattering and UV detectors. So it is possible to use mixtures. It's not that common. You try to not to do that, but if it's necessary for your sample, it's a possibility.
Okay, what is the largest size disparity that can be managed by the technique?
The largest size?
What is the largest size disparity that can be managed by the
the dispersity. So the largest size dispersity. That means a sample that runs from a very small size up to big sizes in one sample. That is one of the strong points of FFF as well, because it covers the full dynamic range. So a sample can have very small components of a few nanometers and include big particles that go up to a micron, and they can be separated in one injection. So again, the dynamic range is the full submicron size range, and a sample can cover this full size range. Then your decision is how good the resolution should be. So the more resolution you want, the longer the run will be. So if there's really a continuous distribution from very small to a micron, the run may be an hour or 1.5 hours to separate the full size range. You are very flexible here. You can also create a method where you have more resolution in certain size ranges of your sample and less resolution in others to minimize the analysis time.
Does the sample have to be characterized before the injection?
That's also a good question. I mean, what we usually do is we use dynamic. If you have a totally unknown sample where you really don't know what the size range is, what we do is we do a dynamic light scattering measurement to just see the size range our sample is in, because that helps us as a first starting method. I showed how you do method development using Scout. There you need to make a decision, what is the smallest size that I want to separate and what is the largest size. So if you have a dynamic light scattering measurement that gives you an indication, you can use that indication and get better and faster to a successful resolution. But you can also, if it's not available or not possible, start with a totally unknown sample, start with a screening method. You may need 1 or 2 more iterations in your flow programming to reach a good result, but that is also possible.
Okay. How different do the particles need to be in terms of charge with the same molecular?
I'm sorry. There's some of the phone line is not that good, so I don't understand some of the words there.
The question is, how different do the particles need to be in terms of charge?
To be separated.
Yeah.
Mm-hmm. Yes. So that depends also on the size. So the larger the size is, the smaller the charge differences we can resolve. The smaller the particles are, the larger the size differences must be. If we think in units of charge of mobility, that is the mobility of the particles, if they differ by something like 0.2 to 0.3 micrometer per volt and centimeter, that would be sufficient to get resolution. What you can also do is, if you can use Scout to simulate the separation. So if you enter a particle size you can go into the software, put in a mobility value for 2 samples, and then Scout will calculate what the expected shift and retention time is. So for a given sample, given size, you have some expectations of what the charge differences could be. You can use the software to tell you what is the expected shift and retention time, and that will tell you if it's possible to measure this difference or not, if it's too small.
Okay, fantastic. I'm afraid that's all the time that we have today. Thank you again, Dr. Johann, for your fascinating presentation, and thank you, participants, for being a great audience. Be sure to check CNN or CNN Online.
Thanks to everybody.
Yes, thank you. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you, Antonio for Technology and Production Services, and thank you, Wyatt Technology, for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Linda Wang. Goodbye.
Okay, thanks a lot.